I am Jay Kaushik, Principal Scientist at National Dairy Research Institute, Karnal, Haryana. Today I will be talking about the two related techniques, circular dichroism and optical rotatory dispersion. They are very closely related techniques, but they are very powerful for determining the structure of small molecules as well as macromolecules like DNA and proteins. So this technique evolved long back, and with the advancement in instrumentation, today we can use it to calculate the secondary structure of proteins, protein-DNA interactions, structure of DNAs like A form, B form as well as G form. Not only this thing, we can also determine how the protein is interacting with another molecule of protein or with the DNA or small molecules. The power of this circular dichroism is basically based on the conformational changes, the asymmetry of the molecule. So when the asymmetry of the molecule is changing, and hence the signal coming out of the machine will also get changed. The underlying mechanism is the molecular asymmetry, and DNA, protein, carbohydrates and many other molecules, they are asymmetric molecules, and hence we can make use of this technique very well to study their conformation, structure, their interaction with other molecules as well as small molecules. So in case of small molecules, it is usually the structural part which we can investigate, while in macromolecules or bigger molecule, it is the conformational part which we analyze with circular dichroism or optical rotatory dispersion. These two techniques have the same origin at quantum mechanical level. So let's see, we will start with this thing with the two objectives that we'll be talking about in next slides. Thank you.
Module on circular dichroism principle is covered under two different parts, part A and part B. In part A, the asymmetry of objects and molecules and the origin of optical activity will be explained. In part B, I have covered linear polarization and circular polarization of lights and their interaction with optically active medium, resulting into the phenomena of optical rotatory dispersion and circular dichroism. In this presentation, I shall be covering concept of molecular asymmetry and optical activity.
To understand the concept of asymmetry, chirality, optical activity, linear or plane polarization and circular polarization, I will show you what is the meaning of symmetry of objects, symmetry of small molecules and macromolecules, what is the meaning of optical activity and how it is related to molecular asymmetry. Furthermore, we shall learn what is the polarization of light. We will also learn linear and circular polarization of light, how a polarized light is produced. So that is the major idea. We shall learn circular dichroism and optical rotatory dispersion phenomena, how CD and ORD can be used to differentiate configuration and conformation of optically active molecules. I shall also discuss the relationship between CD and ORD in this lecture. If you see this slide, we have created some animations to show the difference in circular dichroism and optical rotatory dispersion. The phenomena involving differential absorption of asymmetric lights, that is LCPL or left-handed circularly polarized light, and RCPL, that is the right-handed circularly polarized light by molecules with chiral centers, resulting in an elliptical light in the medium. So what is the meaning of this one? Let's see animation in panel A, that is on the top. You can see two electric vectors rotating in opposite direction. The electric vector shown in green color is rotating in clockwise direction. It is known as right-handed circularly polarized light or RCPL. The other one shown with red color is left circularly polarized light rotating in anti-clockwise direction. We shall discuss the polarized light at greater detail after a while. Now, You can see a box with light orange color placed between the left and right windows on the top panel. It contains the optically active medium. Observe how LCPL, that is the left part, and RCPL, that is the right circularly polarized light, are propagating when they enter the medium. And note the continuous change in ellipticity of the RCPL shown in the green color and when they emerge from the medium. The light shown in blue color is the resultant of superimposed LCPL and RCPL. It is oscillating linearly in the incident light. You can see change in the ellipticity of the resultant light because of absorption of one component, that is the RCPL, shown in green color when passing through the medium. This is what we know as circular dichroism, because there is a differential absorption of only RCPL as compared to LCPL component of the light. The second panel from top shows only the electric vectors at the entrance of the medium. On left side window shown in gray color, and electric vectors of the emerging light after passing through the optically active medium on the right side window. Here the waves traveling through the medium have been removed to show only electric vectors of the incident and emerging light. Now let's see ORD shown in panel B. So what is the ORD? So that is depicted in panel number 3. In this case, I had tried to show only ORD and no CD. What it means that we set some parameter so that it shows only rotation of the light. If you see from the in panel B on left side window, that is the incident light. In the incident light there are two vectors, again shown in red color, rotating anti-clockwise and rotating in clockwise, that is shown in green color, and which is shown in blue color, the bigger one which is oscillating horizontally, is the resultant of left and right circularly polarized light. So what it means is the sum of these two vectors, and you see that the component vectors, they are rotating in opposite direction, but the blue one is oscillating in only one direction, that is the horizontal. So it indicates that if you add left circularly polarized light and right circularly polarized light, they will provide you a linearly polarized light. So when this light, it enters into an optically active medium that is present in the box in between the two windows, when it is passing through this thing, there is a slow rotation of the vector which is shown in blue color. So that is the addition of left circularly polarized light and right circularly polarized light. So if you see it slowly, then the vectors or the resultant vector is slowly rotating downwards, and or you can say it is rotating toward the left. When it has emerged beyond the box wall, there is no change in the left or right circularly polarized light, and hence there is no change in the rotation of the emerging light, which is the resultant of the two. So now what is the ORD? The ORD is when this rotation is taking place as a function of wavelength. What I shown is here only monochromatic wave, that is the wave at particular wavelength. So that is rotating and providing a particular rotation after it has traversed through the medium. But if we keep on changing the wavelength, so this angle of rotation, we call it alpha, convert it further into molar rotation. So that will be different at each wavelength, even for a given molecule or media. So the last panel in this slide, it is just the same as shown in the upper panel in B. So in between this is the chiral medium or optically active medium, and if you see only left panel and right panel in this particular or windows, you will see that the two vectors responsible for electric vectors of circularly polarized light in left-handedness or right-handedness. So they are rotating clockwise and anti-clockwise, and the resultant is again that blue color. That's when after coming out of the chiral medium, the angle of rotation has changed. Previously, before entering into the medium, it is oscillating horizontally, but now it is rotating almost at 45-degree angle from the previous one. So this angle is the rotation angle, and the phenomena as a function of wavelength is known as optical rotatory dispersion. As I mentioned earlier, circular dichroism and optical rotatory dispersion are closely related property of optically active medium. CD is related to differential absorption of light, while ORD is related to birefringence, that is the birefraction of the medium. In an optically active medium, light is absorbed and dispersed depending upon the polarization and propagation direction of the light. It means that differential absorbance of LCPL component and RCPL component leads to circular dichroism, while the differential refractive indices of LCPL and RCPL results into optical rotation of circularly polarized light in an optically active medium. The difference in refractive index depend upon the wavelength of circularly polarized light and hence rotation of the polarized lights. We can say that rate of change of rotation, or more precisely specific rotation or molar rotation with respect to wavelength, is the optical rotatory dispersion. CD and ORD are related optical properties of optically active media, as I mentioned earlier, and one can be derived from another by using the Kramer-Kronig relations. It also means that dispersive and absorptive properties of optically active media are not independent, but can be determined if one of the property is known, because both originates from the same electronic system of the optically active centers. The Cotton effect in absorption wavelength region of optically active medium. Cotton, while observing the optical properties of potassium chromium tartrate in solution in its absorption band, observed that for an optically active medium, the extinction coefficient of LCPL and RCPL components of light were different. So that was the first observation or the recording of circular dichroism. Here it is important to note that this happens only in the absorption band of optically inactive medium. There is no such phenomena. Both the lights are absorbed to the same extent, and hence the difference between the two lights will be zero when they emerge out of the optically inactive medium. Optically inactive medium or mixture of optically active medium do not show differential absorbance of component LCPL and RCPL. So this is the hallmark of only optically active medium. This also means that the electronic system or electronic transitions responsible for the differential absorption of polarized light by the optically active medium. It has also been observed that the molar rotation increases with decrease in wavelength for one isomer of an optically active medium, and this is known as the positive Cotton effect. You can see curves in panel on upper left side. If you see this panel, when we decrease the wavelength, we go from right side to left side. That is the decrease in wavelength. The curve shown in red is increasing. So if such phenomena takes place, then we call it positive Cotton effect. If we decrease the wavelength and optical rotation or molar rotation also decreases with decrease in wavelength, then we call it negative Cotton effect. And two enantiomers of a optically active media will have exactly opposite cotton effects or mirror images of each other. The blue curve in this panel, it indicates the circular dichroism band. You will see that the optical rotation decreases in wavelength, and when it approaches the peak of the CD band, it suddenly starts decreasing and passes through zero at a wavelength at which circular dichroism is maximal. It keeps decreasing further, reaches to the trough, and then again start increasing. So this is the cotton effect. We know it as a cotton effect. And for the isomer of the same molecule for which these spectra were recorded is shown on the right side bottom. So if you see these two, they look like mirror images of each other. It is important to note that the medium exhibits cotton effect only in its own absorption band, and also if the electronic system responsible for the absorption band contributes to the optical activity. The optically inactive medium will not show ORD passing through the zero in the middle of the ORD curve, which I showed you a while ago. There are two kinds of cotton effect, as I mentioned, negative and positive. To remember what is the positive cotton effect, what is the negative cotton effect, let's see how to remember it. We can also say that in positive cotton effect, the optical molar rotation peak is at a higher wavelength than its trough, whereas in the negative cotton effect, opposite is true. The left side panel shows positive cotton effect, while the right side panel shows negative cotton effect. You can also see that absorptive CD band is restricted to a very narrow zone of wavelength, while optical rotatory dispersion or ORD away from the absorption band is still going on. This could have several ramifications in the experimental setup and gathering amount of information. CD band is located where medium have normal absorption band and hence describe the electronic system more accurately. CD signal are more sensitive and precise due to small window of absorption, while ORD is more extended and dispersive, taper down in a shallow manner from the center of absorbance peak, as you can see in both the figures. But ORD spectrum is extended well beyond absorption band that may provide additional information in the wavelength zone where CD measurement is difficult or where CD signal is very strong. LCPL and RCPL will always experience different refractive index at all wavelengths, and hence ORD can be also useful in the region of very small or no CD, but where OD signal is measurable. This is the overall depiction of circular dichroism and optical rotatory dispersion phenomena. In CD experiment, equal amount of LCPL and RCPL is sent to the optically active medium where they are absorbed to different extent and emerge as two still circularly polarized lights but with different amplitudes. The combined resultant light, that is the addition of LCPL plus RCPL, shown with dotted curve in figure on right side, is elliptical and oriented with an angle alpha with respect to plane of incident resultant light. It is there on right side figure between two axes, one vertical shown as solid line while another as dotted line oriented at an angle of alpha. In CD, we report difference in extinction coefficient in LCPL and RCPL or equivalent ellipticity, denominated as theta in terms of degrees. The ellipticity is measured in millidegrees since the difference in absorption of two lights is usually very small. The difference in the extinction coefficient and ellipticity are related quantities and their relation will be shown at later stage. The figures shown are same which I showed in previous animations at the two ends, that is at incident and emergence windows. Panel on left side shows EL, that is the vector, electric vector of LCPL rotating in anti-clockwise direction. It is shown with red color, while RCPL is shown with green color, and electric vector ER is rotating in clockwise direction. If you see figure on left side, both vectors are equal in magnitude but rotating in opposite directions before they enter the optically active medium. The resultant electric vector, which in addition of EL and ER is shown with blue color and oscillating on the vertical axis before light enters the medium. When the two component vectors rotate in opposite direction but equal in amplitude, the resulting vector of the combined light oscillate only linearly in its plane, that you can see in figure on left side, the big blue color vector. It oscillates from top to bottom, passes through the 0. Now, see the panel on right side, which is the scenario after the lights have emerged from the medium. The medium is optically active medium. Please see that EL, which is electric vector of LCPL, has not changed, while the magnitude of ER vector for RCPL has reduced because of absorption by the optically active medium. The electric vector of both the circularly polarized lights are still circular but vary in amplitude, resulting into an elliptical path traced by the resultant vector. If you recall, the resultant vector before it entered the sample was oscillating linearly, but now it is oscillating in an elliptical path traced as dotted lines shown with blue color in right hand side pan figure. Also note that the axis of oscillation has also changed from vertical in left side panel to oriented one with an angle alpha after emerging from the medium. ORD represents the rate of rotation, that is change in angle theta of the polarized light when it is measured as a function of wavelength for an optically active medium. CD is measured in terms of ellipticity of the path traced by the resultant electric vector after it has emerged from the sample. See the minor axis designated as B and major axis A of the ellipse in the panel on right side. The angle theta between the axis of ellipse and hypothenuse of the triangle shown with black dotted lines is the inverse of tan B by A. The angle theta is also known as ellipticity and measured by CD machine as a function of wavelength to record a CD spectrum. In the previous slides, I introduced you to the concept of circular dichroism and optical rotatory dispersions, like how when light rotating in opposite directions, left-handed or right-handed circularly polarized lights, when they enter into the medium, they produce the phenomena of circular dichroism or ORD. Basically, it is the differential absorbance of one component or slowing down of one component in the medium. When they come out, the waveform is different, the resultant of the two component lights. But now we have to understand what is the molecular mechanism or the structure of the molecules, biomolecules, which are impacting the circular dichroism or ORD. So we know it now that the reason behind molecular asymmetry, we had to find it out that what are the causes of this molecular asymmetry and how it is leading to the optical activity of the molecule. The molecular asymmetry is the central point which is leading to the phenomena of circular dichroism and ORD. Now we have to see what molecules or what structures are asymmetric and what are symmetric. Asymmetry provide the optical activity. Symmetry don't provide the optical activity. So we can say that all the structures which are asymmetric may lead to optical activity, but the symmetric molecules, they don't lead to optical activity. So therefore, now we will see what are the molecules which are symmetric and which are not symmetric and which can lead to the optical activity of the molecule. In our day-to-day life, let's see, compact disc is there. This is highly symmetric. A nail is symmetric. A cone, a cylinder or a sphere, they are all symmetric. Why do we say that they are symmetric? Because if you take a mirror image of these objects and try to superimpose over the original object, in that case that the two objects will not be superimposable, then it is asymmetry. If they can be superimposed completely, then they are symmetric. Now, very good example is that you take a sphere, rotate in any direction, any way, upside down, left to right. So the shape and structure of this sphere will not change when you see it through the mirror. The mirror image can easily be superimposed over the original one. So that is this symmetry. This is very high level of symmetry. Similar is a cube. A cube is also highly symmetric. If let's say, but if you paint the cube on all these sides with different color, then it may look asymmetric. So the concept of symmetry and asymmetry is central to the optical activity. Now let's see what are the other objects which are asymmetric. Helix, a impeller, a screw, they have a handedness, left-handed or right-handedness. So we will say that these objects, if we take their mirror images and try to superimpose on the original object, we cannot get a superimposition. They will not superimpose. So such objects which cannot be superimposed are called asymmetric. A very good example is in our day-to-day life we see in the cars. We have two mirrors on both the sides. So left side door has one, right side door has another one. If one is broken, can we replace right side mirror with the left side mirror? So that is not possible. They are asymmetric because they cannot be superimposed. They cannot be replaced with another one. Even in C cells, you have a handedness. Either they are left-handed or right-handed. If you try to put over each other, they cannot be. Or we take the mirror image of left-handed and try to put over the left-handed original image, they will not be superimposable. So such items or structures or shapes, they are asymmetrical. And asymmetrical molecules, so similar concept we can extend it to the molecular structures. So molecular structures which are asymmetrical, we call them in term of chemistry chiral or chirality. So in the next slide we will explore, we will see what are the molecules which are asymmetric, we say them chiral. In the previous slide we saw that what are the asymmetric and symmetric shapes. Now we can extend the same concept to the molecular structures. Now let's see what is the meaning of molecular asymmetry. It can be of two types, structural or configurational asymmetry and conformational asymmetry. In case of small molecule, we can see the structural asymmetry. On left side, all the molecules are symmetric because their mirror images can be superimposed over them. A chiral center is one which is surrounded with dissimilar groups on all the valencies. Let's take the example of tetrachloromethane. Carbon shares bond with 4 chlorines. All are similar and hence carbon in this case is an achiral center, that is symmetric one, because all the groups, all the chlorines, they are similar. Similarly, Benzene is also symmetric since all the carbon atoms make bond with two hydrogens and two similar methylene groups on both sides. Benzene is highly symmetric just like a hexagon. These are regular structures. The alpha carbon in glycine is bonded with carboxylic, amine and two hydrogens and thus is not a chiral center, while all other amino acids where one of the hydrogen on alpha carbon is replaced by a carbon side chain are chiral center. On the other side, we see some example of asymmetric molecule with chiral center. So all the objects or molecules shown on right side, they are asymmetric and have a chiral center. In 1-bromo, 1-chloroethane, all the four groups around central carbon are dissimilar and hence the mirror image cannot be superimposed over the original molecules. Therefore, this carbon is chiral center. Similarly, except glycine, the alpha carbon in all the other amino acids, rest of the 19 amino acids is chiral. All the molecules with chiral center possess optical activity in the region of absorption band, which originated from the electronic system of the chiral center. In macromolecules like DNA and protein, the origin of asymmetry is conformational. That I shall discuss in circular dichroism in practice in another lecture. In the next few slides, I shall explain how plane polarized light, circularly polarized lights are produced, as well as how they interact with optically active medium in greater detail.
You are required to refer part B of the module on circular dichroism principle to understand the phenomena of optical rotatory dispersion and circular dichroism.